So good morning, everybody, and welcome to the May edition of the IIDR Rounds. I'm Laurie Burrows. I'm the Associate Director of the IIDR, and I'm delighted today to welcome our two speakers. So our first speaker is Aisha Nazli, who is a research scientist in the Kaushic Lab. Her research focuses on different aspects of host pathogen interactions and host immune responses against several viral pathogens, including HIV and herpes simplex. Today, she's going to be talking to us about the immune mechanism underlying the preferential transmission of the HIV R5 strain in the female genital tract. Our second speaker will be Dr. Tim O'Shea, who's an associate professor in the Department of Medicine at McMaster. He's an infectious disease physician and the medical director of the Hamilton Social Medicine Response Team. Today, he'll be talking to us about combining addiction medicine and HIV treatment and prevention to increase patient engagement. So thank you to both of you, and I will turn it over to Aisha.
Thank you, Laurie. Thank you for the nice introduction. So good morning, everybody. Today, I'm going to talk about HIV transmission by two HIV variants, X4 and R5. These variants differ in their usage of co-receptor on the cell surface. HIV epidemic still persists throughout the world despite the availability of effective therapeutics. According to WHO, there are 38.4 million people living with HIV worldwide. Among adults who are living with HIV, there is a clear gender disproportionately, and like women are affected with HIV, and they are burdened, and they are almost 54% of women living with HIV as compared to men. It's important to study about the preventative strategies for HIV transmission in women because HIV is the leading cause of death for women of reproductive age. The major mode of transmission in women is heterosexual contact. And this heterosexual transmission of HIV leads to female genital tract where the infection happens. So if we study about the female genital tract, we can divide into two parts, the upper female genital tract and the lower female genital tract. They differ anatomically and differ also in the epithelial lining, these two regions of the female genital tract. In the upper region, it's the uterus, fallopian tubes, and ovaries, which are lined with a single layer of columnar epithelial cells, which are tightly bound together with tight junctions. The lower female genital tract is lined with multi-layer squamous epithelium. So epithelial layers are the first line of defense of any, and they encounter any pathogen entering through sexual route. And HIV has to cross this epithelial barrier to infect the target cells present underneath the mucosal surface. So when, despite these both X4 and R5 variants are present in the body fluids like semen and vaginal fluids, only R5 is detected in the initial stage of HIV infection, and it's considered as a transmitter virus. So what are the factors which are stopping X4 variant from causing the infection and blocking it on its initial stage of infection? So scientists reported that immune cells are the one that restrict X4 to establish infection, but there is a gap in literature to see if there is a role of epithelial cells for acting as a gatekeeper in selecting R5 transmission. in Kaushic Lab, we use ex vivo model to study the initial stages of HIV infection. The way we isolate these cells is described in the diagram underneath. And like, I'm not going into details because of the time restriction, but if anybody is interested, we have already published this method of isolating primary genital epithelial cells. In briefly, I can describe that like we collect the tissues from pathology and like we digest them, filter them, and we isolate these epithelial sheets, which we seed on these transwells, and they grow in confluent monolayers. So using these ex vivo model of primary genital epithelial cells, we try to recapitulate the clinical scenarios where R5 can transmit the infection. So we added these confluent monolayers with fluorescently tagged virus. R5 was tagged with DS red and X4 was tagged with GFP. And when these viruses were added on the apical side, we collected basolateral supernatant and added to the uh, indicator cell lines. The images below, you can see the fluorescently infected cells. And mostly we notice that R5 is translocating through these genital epithelial monolayer. So we can conclude from these observations that R5 can translocate through genital epithelial monolayers efficiently, which suggests that there is a role of genital epithelial cells in the R5 transmission. So where is the X4 virus? In addition to the basolateral supernatant, we also collected the cells which were exposed to these viruses, and we lysed these cells by freeze thawing. And these cell lysates were added on the indicator cell lines, and we noticed that all the X4 virus was trapped intracellularly in the genital epithelial cells. So this means that like genital epithelial cells have the ability to trap X4 virus while letting go R5. So this observation leads us that like we should try more than one strain of X4 variant and R5 variant to confirm our finding that it's a universal phenomena among all R5 variants. So we use different strains belonging to X4 variants and R5 variants. And at 4 hours, we fix the cells to see if the viruses are entering into the genital epithelial cells at the same rate. And you can see here in the graph that almost all the viruses were entering into the cells at the same speed. At 24 hours, there was a completely different story. We saw that most of the X4 virus was trapped inside the early endosome, which is stained red here, and the virus was turquoise in color, is stained turquoise. And when these two co-localize, it appears as white. So you can see in the graph, when we measure the fluorescence of virus present in these cells, we found that X4 was still trapped after 24 hours, while most of the R5 was gone through these cells. So what are the potential antiviral pathway in genital epithelial cells that can trap X4 virus? So previously, a few years back, we published one of the paper where we mentioned about that HIV envelope protein GP120 can activate TLR2 and induce interferon beta through that TLR2 pathway. So we wanted to look that TLR2 has any role in trapping the virus. To look at TLR2, we stain the cells after 24 hours after exposing them with X4 and R5 strain. And we found that X4 upregulate TLR2 expression, which you can see here. The cells were also stained with EEA1, which is an early endosomal marker. And we can see that like TLR2 is co-localizing with EEA1, which is an early endosomal marker, which indicates that X4 not only upregulates TLR2 expression, but also activates uptake of TLR2 in early endosome along with the virus. So TLR2 signaling has two sides. On the plasma membrane, it can activate different pathways, while on endosomes, it can activate type 1 interferon. So we looked at interferon beta production by these two variants, and we found that X4 and R5, both strains, can induce interferon beta as compared to mock controls and the cells which were exposed to envelope deleted mutant. But X4 can induce much more significant amount of interferon beta as compared to R5. So we took a deeper dive into interferon signaling pathway. And as you all know, that type 1 interferon, when it is produced, it can protect the neighboring cells by interacting to IFNAR, the receptor, and it can activate interferon stimulated genes. So we looked at 10 different interferon stimulated genes or ISGs by qPCR to see the gene expression of these ISGs after exposing to X4 and R5 viruses. And we found that although both induce somewhat upregulate the expression of these ISGs, X4 induce much more higher ISGs as compared to R5. So we concentrate on like ISGs which are responsible for blocking viral release from the cells, and BST or Tithrin and ISG15 are the two ISGs which are known for their function to block the release of virus from the cells. So we looked at these two ISGs, and we noticed that X4 induce not only mRNA expression, but also induce protein expression of BST2 and ISG15. These two ISGs are known that they literally tether the virus on the cell surface. So we wanted to see if we block these two ISGs, what will happen? So we noticed that if we block these two ISGs by using siRNA, we significantly reduce the amount of intracellularly trapped X4 virus. When we combine the siRNA for BST2 and ISG15, we completely release the X4 virus from getting trapped inside the cells. So if I want to summarize my results, I will say that like X4 binds to its co-receptor on the cell surface, increase the expression of TLR2, get into the early endosomes, and it also initiates the endosomal uptake of TLR2, and activates TLR2 signaling in the endosomes, which activates interferon beta signaling pathway and induce ISGs. Two of these ISGs are responsible for blocking X4 release from the early endosomes, and it completely trapped the virus inside the early endosomes. While in case of R5, it goes through transcytosis and exocytose from the cells very quickly, escaping all the immune recognition and activation of all the signaling pathways. So I want to acknowledge my Kaushic Lab, and especially Dr. Charu Kaushik, for all the encouragement and guidance in this project. And I also want to say special thanks to the women who generously donated their tissue for this study, pathology staff who helped me getting those tissues, and healthcare staff and nurses in the same-day surgery for their assistance in taking consents from the patients and the funding agency. Thank you for listening. Any questions? I will be happy to answer the questions.
Thank you, Dr. Nazli. I think we're going to hold the questions until the end after Dr. O'Shea has a chance to talk. So if anybody has questions, please type them into the Q&A and we'll answer them at the end. So we'll pass it over to Tim.
Good morning, everybody. Thanks for giving me a venue to talk about something that I'm quite passionate about, which is a clinic that we've been running, providing a number of different services, but started primarily as a HIV treatment and prevention clinic in the downtown core of Hamilton. And I'm going to talk a little bit about some preliminary findings from our clinic evaluation. All right. So in terms of background, so there's lots and lots of literature that documents the fact that the healthcare system lags behind in its ability to care for people who are at risk of HIV infection or those who have established HIV infection, who have concurrent substance use disorders. And that is compounded, the failure of the healthcare system is compounded when there is concurrent issues of homelessness or issues of mental health disorders. I quote one study here, which was just recently published in April in the journal AIDS, where they looked at a cohort of patients in Vancouver, and homelessness status was associated with a 44% decrease in odds of progressing through the overall HIV care cascade from diagnosis through to treatment and viral load suppression, and between 41 and 50% decrease in odds of receiving therapy, being compliant on therapy, and achieving viral load suppression. There's other studies that document that when care for HIV is combined with things like addiction medicine services for people who use drugs, there is an improvement in some of these measures, but the numbers still lag behind the general population. This is a graph from one of the studies that looked at engagement with opioid substitution therapy, and comparing that to patients who use drugs who have HIV who aren't on opioid substitution therapy, and how they do in each step through the care cascade. So you can see that as you move through the care cascade, people fall off in both groups, those that are on opioid substitution therapy being the darker bars, and those that aren't being the lighter bars. But there's a clear discrepancy between the two at each stage. And if you get down to viral load suppressed, those who aren't on therapy have only a 42% chance of having their viral load suppressed. So we started our project back around the fall of 2019. And at the time, there was a bit of an epidemic and spike in the number of people in Hamilton who had a new diagnosis of HIV, who documented injection drug use as one of the risk factors for acquiring their infection. And this rose to a historic high in our city of 44% of new diagnoses in March of 2020. That prompted a bulletin from Public Health outlining their concern over this trend. and asking for people to kind of be aware of it, although there wasn't much in terms of what to do about that. At the same time, we know that overall in Ontario and Canada, but particularly in Hamilton, opioid-related deaths have continued to rise over time. And hepatitis C infections as well are higher in the cohort of people who use drugs in Hamilton than the provincial average. We know as well, again, just from clinical experience, and this is not a unique thing to Hamilton, but again, across different settings, that the group of patients who we're talking about here, again, the confluence of opioid use disorder, homelessness, and mental health issues are traditionally poorly served by the general medical system, and that ends up being a huge barrier to care. This just shows again, which I think if anybody's following the news, they would not be surprised to see that opioid-related deaths in Hamilton have really continued to rise quite dramatically. This is an ongoing daily issue in our city. It's a huge use of healthcare resources and a huge cost in terms of loss of life and ongoing trauma for the community of people who use drugs. So looking at these two issues, the group that I work with wanted to see if we could do something about it. And so what we did was we talked to the community. We're well linked with an organization called Keeping Six, which is a group of people who use drugs that work together as an advocacy group. And they helped us, and as well some of the patients that we work with as well helped us to try to decide what would be an effective intervention to try to deal with some of these issues in our community. What we heard fairly loudly was, again, a main theme really being a deep distrust of the medical system. And that distrust is well earned through multiple episodes of trauma that people have undergone, as well as that their friends have undergone. So that the people that we were trying to reach were really distrustful of anybody who identified as part of that system. The second part was the extreme difficulty of trying to engage in care for medical issues in the setting of trying to stay alive when somebody doesn't have a home, doesn't have food security, is dealing with poison drug supply, and just trying to exist on a day-to-day basis in that setting makes it extremely hard to access care in a traditional clinic setting. And then the other main issue that came up again and again was the fact that most people that we were trying to reach and engage in care identified their substance use as their primary issue. And that was the thing that took up most of their energy on a day-to-day basis, was trying to stay out of withdrawal, trying to figure out how they were going to access the drugs that they needed to stay healthy. And so the intervention that we came up with is the one that I'm going to describe in the talk today. The first step in designing this clinic was to find a place where the community that we were hoping to work with felt comfortable, felt welcomed, felt safe. And we heard over and over again that the AIDS Network in downtown Hamilton was such a place. The AIDS Network is an AIDS service organization that's been operating in the city for many years and has established a trusting relationship with the community of people who use drugs. They provide harm reduction supplies through a harm reduction room that gets accessed multiple times throughout the day. They have harm reduction workers who have excellent relationships with the community. They provide things like drop-in lunches and that type of thing. And again, it's just a place where people generally feel welcomed and safe. They were kind enough to offer us some space within their organization where we could run a clinic. And so we set that up initially, like I said, in 2019. The clinic is run by an organization called HamSmart, which is a group that I'm part of, the Hamilton Social Medicine Response Team. We are a group of healthcare providers and community organizers who work together to integrate clinical care, critical analysis, and political action to try to improve the health and well-being of our patients. We run a clinic now at the AIDS Network, and it's grown over time from 2019 to today. But where we sit now, we have a doctor, we have a full-time RN who's in clinic every day. There's a clinic administrator who greets patients at the door, a program manager who is a person with lived experience of drug use and homelessness, and peer support workers who are available to support patients in the clinic space. So the clinic, like I said, operates out of the AIDS network. Right now, we run 2 clinics a week. We run a Tuesday clinic that goes from 10 o'clock in the morning until 4 o'clock in the afternoon, and then a Thursday clinic that runs from 12 till 4. Primarily, the service is intended for patients who are rostered under our care, but we almost always have about a quarter of the patients just walking in, seeking care as well. So we do provide some drop-in care. We offer both primary care, HIV care, as well as addiction medicine care. And the name of the clinic that does the addiction medicine is the Support and Safe Supply Clinic. I'll talk a little bit about Safe Supply in a second. Drop-in care can include anything really. So the number of different sort of issues and complaints that we deal with on a day-to-day basis is quite wide. The clinic itself is, I think, a pretty neat place to be. So just describing our Tuesday afternoon clinic. So the space, if you sort of came on a Tuesday afternoon, is always extremely busy. There's lots and lots of people around. The picture in the top right-hand corner of the screen is myself and Claire Bodkin, who's the co-medical director with me in our clinic room. We have drop-in psychiatry care on Tuesday afternoon. So Dr. Rachel Lamont comes on Tuesdays and sits in the back room, and people can just walk in and see her without an appointment. She's extremely busy because word has spread that she's available, and lots of people are coming in to see her on a Tuesday afternoon. Keeping Six, as I mentioned, is an organization of people who use drugs that operates a drop-in lunch on Tuesday afternoons in our space as well. So on a typical Tuesday afternoon, we serve between 50 and 80 lunches. The lunches are extremely, they're really quite good. The person who cooks is a person with lived experience of drug use who used to be a professional chef or still is a chef. And so he prepares extremely excellent meals for everybody who pop in. We have we have the AIDS network running their harm reduction room. So people are kind of coming in and out and getting safe supplies from the harm reduction room as well. So generally through the doors on a Tuesday, there'd be about 100 people coming in and out. And of those in the medical clinic, we would see somewhere in the range of 20 to 30 between 2 physicians doing addiction medicine and drop-in care, and then the psychiatry service as well. Just wanted to quickly mention safer supply for those who might not know the paradigm that refers to. So I mentioned at the start the data that shows that people engaged in opioid substitution therapy do better in terms of their HIV outcomes. We know again from studies as well as just from knowing patients that some people do extremely well on traditional opioid substitution therapy and some people don't. And safer supply is really an intervention that's designed to help prevent overdose risk primarily in people who have not been able to stabilize on traditional opioid substitution therapy. What it means exactly is that we provide pharmaceutical grade opioids to patients, typically hydromorphone tablets that they pick up on a daily basis at the pharmacy, and then they use as they see fit. So some of my patients swallow all of their pills, some of my patients smoke their pills, some of my patients crush and inject their pills. All patients receive instructions on how to do these things safely. And the idea being that accessing a pharmaceutical grade opioid is going to be much safer from an overdose prevention point of view than accessing a tainted drug supply on the streets. We don't have a lot of data around the efficacy of safer supply. The biggest study that we do have came out of London, Ontario last fall. I'll highlight a couple of the results that came from that. This is their main summary table where they looked at patients who were enrolled in the Safe Supply program 1 year before their entry and 1 year after, and compared a number of different variables that you see listed on the table. And then they also looked at a matched unexposed cohort who were unexposed to Safe Supply, but had similar characteristics at baseline to the patients in the program. And essentially what you see is that for most things, there's fairly impressive improvement for patients in the Safe Supply program that is better than the matched unexposed individuals. Looking at other factors that have been examined in Safe Supply programs, in addition to the health benefits, we see that patients have increased engagement in primary care. Importantly for this talk, increased engagement in HIV care, increased access to housing, and decreased involvement in the criminal justice system when they're enrolled in a safer supply program. So all of that to get to what is really a quite simple evaluation of our clinic model, which we've been undertaking over the last couple of years. And again, it's an extremely simple research question that says, is a clinic like this that tries to lower barriers to care and provide a more patient-centered experience effective in increasing engagement in care as well as retention in care for HIV? It's a mixed method study, and I don't have all of the results yet because the initial analysis is ongoing, but we did some quantitative and qualitative analysis. We looked at all patients in our clinic who were HIV positive or at risk of HIV infection. And we, again, just wanted to see whether or not this type of a model would increase their engagement in care and retention in care. Long story short, it's a very small sample size in our initial analysis, but it seems like it does work. Our initial study sample is 13 participants, all of whom had HIV infection. Our retention in care was 100%. So over the time period that we were following patients, all 13 made all of their data points in terms of our data collection and were retained in care at the end of the study period. Important to note that this was over the COVID-19 pandemic period. We did lose access to our clinic space for a number of months during that time, but we were able to maintain contact with our patients through other means. Of the 13 participants at the end of the study period, 11 of the 13 had an undetectable viral load. And then just looking at a secondary kind of outcome for us in terms of their opioid use disorder, this is all self-reported. Patients reported reduced cravings, reduced fentanyl use, and reduced injection use. And the take home from that is that there's a reduced risk of overdose. We haven't had any overdoses in our clinic population. So preliminary findings again, but I think that they do point towards a potential promising model of care for people who use drugs who both have HIV infection, but also who are at risk of HIV infection. And just in terms of summaries of what I think is effective about the clinic, and some of this is reflected in our qualitative data as well. The first aspect, again, is that we try to be as low barrier as possible. We generally don't book appointments. People can drop in at any time that works for them. We have set clinic days, as I mentioned, Tuesdays and Thursdays, but our RN is there in the clinic space 5 days a week. And so if people do drop in at a time when the physician's not there, Amanda's able to see them, figure out what they need, call one of the doctors. We have a 7-day-a-week on-call line, and we can kind of deal with their medical issues at that point in time. I think, again, very key to the initial success and the ongoing success of the clinic is that it's co-located in a place where people already felt comfortable, where they felt safe being. And we sort of piggybacked on that trust to establish a relationship with patients, and then we were able to kind of build up a trusting relationship over time. I think, again, that it's important for us to remember as a clinic that we're there at the invitation of the community. And so we're sort of the guests in the space, which I think also works in terms of some of the power dynamics that typically exist in the medical field and can offer a bit of a barrier to care. Thirdly, we do our best to be led by what the patients identify as their primary issues. So again, for most of our patients, that's substance use and lack of access to medications that help them to manage their substance use disorder. And so we try to be nimble in terms of what we offer. Many of my patients are on traditional opioid substitution therapy and do well on that. But those that don't, we offer safer supply prescribing too. All of my patients at this point in time are on some form of daily dispensing medications as well, which works quite well in the paradigm of HIV care because I dispense their HIV medicines at the same time. They're motivated to go to the pharmacy to get their opioid substitution therapy, and they pick up their HIV meds at the same time and take them there at the pharmacy. We also note that, again, for most of the time that I'm spending with patients isn't around their HIV care at all. It's mostly around their substance use disorder and pressing needs like housing and the need for a tent or a blanket or a sleeping bag, making sure that they have food that they can eat and those types of things. So that tends to be the majority of the care. And then lastly, we are very much a peer-led organization. So I think in most traditional settings, the physician is seen kind of as the leader of the clinic or the boss of the clinic, or at least a main decision maker. maybe that's not always true. Maybe some of the physicians would disagree with me. I don't think maybe they always feel like the boss. But what I will say is the patients aren't often seen as the boss. And in our clinic, it really is led by the patients in terms of what types of services we offer, how we offer the services. People with lived experience are involved in every decision that's made about how we run the clinic. The peer support workers are part of every discussion in terms of how we're approaching kind of difficult situations or what to do about, problems that arise. And so again, making sure that we're taking the lead from the community, I think means that we're offering a service that's acceptable to that community. So next steps and where we go next with this. So our initial analysis, as I said, was 13 patients. We're now up to 22 patients with HIV infection within the clinic. There are other clinics doing similar work, probably across Canada, but we've established links with similar clinics in Ontario and Quebec, and we're hoping to develop some larger cohorts to follow along and offer a bit more robust evidence. Continue to work on addressing remaining barriers of care. So I think that we continually are trying to identify who isn't being served in our clinic. One of the issues that came up there was that oftentimes people who identify as women aren't feeling comfortable in a space where there's lots of men. And so we've started just in the last 6 months offering a clinic at the YWCA, which is for women, people who identify as women only. And that operates in concert with their safe use space at the YWCA. We also have patients who just don't feel comfortable coming to clinic or can't get to clinic at all. And so for those patients, we're continuing to build an outreach service that will go to them and meet them where they're at. We want to expand our offering of PrEP for people who use drugs, both to offer protection to the community as well as to start to develop some evidence base around that. And then just expanding our overall clinical services and making sure that we're doing a good job of making sure people are treated for hepatitis C and other STIs as well. So acknowledgements, again, Keeping Six is an integral partner in this project and continues to kind of help us inform how we're doing things. The OHTN is the funder of our initial analysis and ongoing clinical evaluation. I mentioned HamSmart, the Department of Family Medicine at McMaster is providing the research infrastructure for the project. And then the AIDS Network is our partner in terms of our location. And that is all for me. So happy to move to questions. Thank you.
Thank you very much. So I will open the floor for questions. So there's a question in the chat for Dr. Nazli from Jake. He asks, do you have any hypotheses as to the viral factors that differ between X4 and R5 that might be driving TLR signaling by X4 and not R5?
So the difference between X4 and R5 is the envelope protein, and that's how they use different co-receptors. So my hypothesis for this is that this is the GP120 structure of envelope protein, of X4, which is guiding TLR2 and activating TLR2 signaling, and that's how it's happening. If we try to change the envelope proteins to R5 on X4 backbone, we will see completely different story. So this is totally depends on the envelope protein, how it activates all the signaling pathway in the cell.
Mm-hmm. I had a sort of related question, which is what is the history of how these lineages appeared? Like, are they from different geographies in the world, or is it a response to treatment, like some kind of escape mutant?
So this usually R5 is in the initial stage of infection, and because of mutations, it can, in chronic stage of HIV, it can change into X4 lineage. And so this initial stage is important, that how it escapes the immune system and like how it don't get recognized by the immune system.
Okay, thank you. So there is a question for Tim from Kevin. He says, have you started to look at the possibility of using injectable PrEP for HIV prevention?
Thanks, Kevin. Good question. Probably you and I should have a coffee again sometime to talk more about PrEP, because it's something we haven't really done a great job of getting going. Interesting around the injectable side of things, I think the main benefit of injectables, both as treatment and as PrEP, is the issue of compliance. Again, for many of my patients, compliance hasn't been an issue because they're, like I said, going to the pharmacy every day and picking up their medication. So I think that there's some patients that would be very helpful for, because they just would rather not take a pill every day. But for the most part, and again, this is mostly my experience on the treatment side, people have preferred to stay on oral therapy rather than injectable therapy. Partly because there's some needle phobia, which is always interesting to me in my patient population, but also because they're going every single day to the pharmacy to pick up their meds and they're getting them anyways.
Thank you. And I think the next question's similar from Marie Eve, which is, what are your thoughts on long-acting injectables to care for that patient population?
Yeah, same. So I think there's people that aren't engaged in care. I think it makes a ton of sense for people that aren't necessarily going to get opioid substitution therapy every day at the pharmacy. It's a huge benefit too. Of my 20 however many patients I had down there, there's only 2 currently that are on injectables, and that's that patient preference. But definitely, I think, again, if I was to sort of think of the patients that we're missing at this point, we're missing people that aren't interested in any opioid substitution therapy at all, and so they're not engaged in care at all. And I think that group, there's definitely promise to injectables.
I found this model fascinating. I think it's a fantastic idea. I was wondering, you were talking near the end about reaching out to people who still have barriers, right? So setting up a parallel clinic for people who identify as women and doing outreach to people who can't get to you. So I know you have, sort of some block of funding to carry out this type of work. So where does the extra money come from when you start having to add on these modules that are different from what you're already doing?
Yeah, so right now we're funded a couple of different ways. We did get funding from Health Canada through their substance use, I can't remember what the last 2 letters are, the SUAP program. That is likely renewable. It's renewable yearly for up to 5 years, and then we have to figure out what to do at that point. So we have some funding that way. The other piece that's been very helpful for us is, I think, because of the type of work we're doing, other organizations have been very happy to partner with us. So like I mentioned, the AIDS Network just provided a space to us. We don't pay them. The YWCA is very happy to have us there. They provide space to us. So in that way, we're a little bit lucky in terms of the partnerships. But I think the sustainability and the funding model kind of in the long term is an ongoing question that we are continually working towards figuring out.
And I noticed from the data that you have so far, from an infectious disease perspective, that there's a lot less infections due to injection-related issues, right? Which is, I think, the cost savings is really there.
Totally. And I think, again, the way that I came to Safe Supply, because addiction medicine community in general is a little bit wary, I think, of Safer Supply. And I was a little bit nervous about it, to be honest with you, to start. But I started just in patients who had HIV infection, and it made a lot of sense to me, because having people having their viral load suppressed in a population of people that was generally unstable, potentially sharing needles, made a huge public health sense to me. So if I could kind of say, okay, here we go, we're going to manage your opioid substitution, that's your priority, my priority is managing your HIV, for sure there's cost savings overall, because again, that group of people just isn't spreading their infections, isn't really accessing
well, not landing in the hospital with injection-related infections that are going to cost a fortune, right? I love this set of talks. It just shows the breadth of what we do at the IIDR, from very basic science to community outreach. Love it. Let me go back to the question. There's a couple more. So a question from Merrick for both of you. R5 blocking drugs could potentially prevent mucosal transmission and are not used much in treatment. Would R5 receptor blockers prevent HIV in people who use drugs, or would bloodborne infections enable, I think he means X4 or R5, I don't know, transmission between patients?
I have no
Can you guys
idea.
chat?
So, Aisha, please.
Yes. So the question they ask is about like the HIV transmission of R5 and X4 in injections, or so it is already known. There are many publications which talk about how immune cells, they pose a restriction when X4 virus is try to initiate the infection, but and R5 gets successful. So they both variants are present in blood and body fluids. So they both are transmitted to a recipient, but R5 is mostly the first one to start the infection.
So I guess the question is more about would drugs that specifically block R5 be useful? Okay.
Yeah, on the initial stages of infection, in acute phase of infection, yes, it will be useful to use those, but like to prevent further or to maintain or to suppress the viral load, it will be hard to use those medications without anything for X4 virus.
Okay. All right, great. I don't see any more questions coming in, so I would like to thank both of you very much for speaking with us today, and thank our audience for joining us, and we will see you in June. Thanks, everyone.
Thank you.